And it's a pleasure to introduce Doostmohammadi, who's going to be talking today about engines of life, self-pumping and bio-inspired fluids. Amin got his PhD in mechanical engineering in 2014 from the University of Notre Dame, and then moved to the University of Oxford, where he was a postdoc for I think the first 3 years, and then for the last 2 years, so he was there for 5 years, he was a Royal 1851 Research Fellow in Theoretical Physics, I think in Julia Yeoman's group. And then in 2019, he was appointed as an assistant professor at the Niels Bohr Institute, where he heads the group on active and intelligent matter. So Amin, please take it away. Thank you.
Thanks a lot, Rich. It's a great pleasure to talk about some of the work that we have been doing recently. And as the title suggests, I'm going to talk about engines of life and the idea of self-pumping fluids, which might sound a bit strange as to talk about fluid that pumps itself, but as odd as it might sound, then nature has found ways of doing it. And this is now studied under the context of active matter, and beautiful examples of microscopic active matter are these bacterial swarms or cellular tissues. And what they have in common is that they are intrinsically driven far from thermodynamic equilibrium. This is because we have local energy injection, because of the individual particles work like engines. They take energy from the environment and then they convert that energy into motion. And by doing so, they are able to drive the system continuously and create patterns on the scales that are much, much larger than the scale of the individual. For example, here, looking at the bacterial system, each bacteria is typically the size of a micron, but if you look more closely, you can see there are swirls and jets on the order of 50 micrometers. So this collective motion is one significant property of these active materials. What is fascinating is that we can go even one scale down. We can go to inside of these individual epithelial eukaryotic cells. Here is a epithelial cell. And if you look inside this cell, the cytoskeleton of this cell, this is also an active material. It is constantly taking energy from the environment, converts that into motion, and by doing so, it basically self-propels the entire cell forward and results in the morphological changes and the motion of the cell. Now, what is interesting about these natural active systems is that if we understand how they work, then we can come up with ways of imitating the nature. And this is a perfect example of this, is basically taking some of these subcellular filaments, here are the microtubules from inside the cell, and then mixing them with kinetic motor proteins. As these motor proteins walk on these filaments, they create motion on them. And if you then in the lab put many of these together, then you end up getting structure like this. So it is again continuously being driven out of equilibrium by individual components, and then it's able to create these collective flows. Now, the fascinating thing is that once we have these kind of more simplified systems, then we can have ways of controlling them. For example, we can put these under a confinement. We can put it in a square channel. And when you do that, it just spontaneously self-organizes into a shear flow. We can make it even more interesting if you put it now in a racetrack geometry like this, then it starts flowing. There's no external pressure gradient, it just flows because of the individual components are exerting energy continuously. Now, what is interesting about these kind of flows is that these now have been observed also in 3D channels, and they achieve speeds of about millimeter per second, and they persist up to meter scale, which is actually comparable to the current microfluidic technology that is driven by the pressure gradients, for example. The other amazing thing about this is that it is not specific to these systems. It has also been observed in different systems, in bacterial systems put into confinement, or even the cellular systems, the eukaryotic cells being put under confinement. So this very brief introduction brings me to the main idea of why we are studying these active materials. And to me, this can be put into two general categories. So one is that we are using these ideas of the active matter and active fluids to gain understanding on the physics and mechanics of actual living systems. For example, we know now that in human, when cancer invades, in more than 90% of the time, it does it collectively. The other beautiful example is during the organ development or morphogenesis. Here you can see a fly embryo at the early stages of the formation. Each dot is basically a cell nucleus. And after a while, we can see these cells, they self-organize their flows into these beautiful stripy patterns. And then each of these stripes would go on and form different parts of the fly body. So again, understanding how these collective flows emerge would help understanding these kind of biological problems. But then the second category that I will actually spend most of my time on today is learning what happens in nature to design our own materials that are capable of self-organization, self-healing. People have tried, for example, putting gears inside bacterial baths to direct the rotation of these gears, or making these beautiful self-deforming spindle-shaped droplets out of active materials that I will also talk about. And essentially, my talk is structured also based on these two broad categories. In the first part, I will give two examples from bacteria and from eukaryotic cells that how using active matter we can understand actually things about biology of these systems. And then in the second part, I will focus on emulating nature in order to control these active materials. So the first example I want to talk about is about bacterial competition. This is a collaboration with Oli Mika, Kevin Foster at Oxford Zoology, and Mac Durham at Sheffield, and with Julia Yeomans with the theory part. And the idea is to look at the competition between two types of bacteria, where one of them moves faster. The system of interest is this Pseudomonas bacteria. This is very infectious, nasty bacteria that basically moves by grabbing on surfaces. It has these legs that are called pili, and then by using these pili, it propels itself forward on surfaces. So what they did in the experiments is to create a mutant of these bacteria that basically has more legs. And it moves, when you check in the experiments, the individual bacteria moves 2 times faster compared to the wild type. So I'm going to call these wild type and the mutant. So the mutant moves 2 times faster, and then we ask the question, what happens now when we put many of these together? Does this moving faster give any advantage? So the experiment is set up like this. We have a racetrack between top, we have the slow moving wild type bacteria. Again, these are individually moving slower compared to the mutant. And then we look at who wins the race. And the results are quite fascinating. Initially, the fast moving one takes over, but over long run, the wild type, the slow moving one, is actually has an edge and wins the competition against the fast moving one. We asked if this is an artifact of having these two in separate racetracks. What happens if we now mix these two together? So here you see an experiment where we have the fast moving colored by purple and the slow moving by gray, and the same dynamics happens. Initially, the fast moving one takes over, but over a long time, you see the front gets dominated by the gray slow moving bacteria. And indeed, we notice what happens is that the fast moving bacteria, they get stuck behind in some structures that we call rosettes. So what we wanted to understand is that how do these rosettes form, and why do they form only for the fast moving bacteria? To do this, we looked more closely at this bacterial system. In addition to showing these collective patterns of motion, if we look at the orientation of these bacteria, as it's noted here, we can see that they self-organize into domains of certain size with a certain orientation. More interestingly, where different domains of different orientations meet, there are singularities in the orientation, which are known as topological defects in studying liquid crystals. So just to give a very quick explanation of these topological defects, and apologies to the experts. So in a 2D system like this, in the orientation field, we can have two types of singularities in orientation. These are known as plus half and minus half topological defects. You can think about a comet shaped and a trefoil shape. And these are basically defined based on the topology. If I take the center of these defects, and if I take a 2 pi rotation like this, you can see that the orientation of the particle basically rotates by pi. So over 2 pi, the orientation rotates by pi. So this has a plus half charge, while if I take this 2 pi counterclockwise rotation, the orientation of a trefoil rotates for minus pi. So this has minus half charge. And these defects, these are basically characteristics of any system with orientational order. We can have other types of these, like a aster or vortex that have plus 1 charge. And you can see this all over the place. One very common example is in our fingerprints, where you can actually see these plus half and minus half defects. And also many of us, we have these plus 1 defects also on our head. But that's because of the topology of the head that imposes this kind of full integer charge defect. Anyways, the important point that I want you to take from this is that by looking at these patterns of orientation and seeing these two types of defects, then we can conclude that this system is behaving like a particular type of liquid crystal known as nematic liquid crystal. But it is not quite like the classic liquid crystal in a sense that the system is active. If you look at the dynamics, you see that these defects, they constantly get produced, and also constantly they annihilate each other. When defects of opposite charges come together, they annihilate each other. So the first step for us was to make a simplest possible model that we can use to look at this system. And the simplest model we could think of was basically considering a bunch of hard rods that are interacting with each other through volume exclusion. And each rod has a force on it, as shown here. So a couple of Langevin equations describe the system, and qualitatively, it creates the same patterns. But we can do better than this. We can now take these defects and look at the velocity field around these defects, and in particular, average velocity field around these defects as they move around. So here on the left column, you can see the experiments. On the right, you can see the discrete model. And you can see essentially the discrete model picks up the patterns of the flow field that you see in the experiments. And the clear difference between the flow patterns of these two defects, which I will come back to in a short while. At this point, we asked, okay, in addition to this, can we also have some kind of continuum description that we can look at the same flow fields from the continuum point of view? And we adapted the equations of active liquid crystals, in particular active nematics, to look at this. So to do this, we need to first describe the order parameter corresponding to the orientational order of these particles. This is governed by this type of tensorial equation. So we have a Q tensor which characterizes the orientation of particles. It is a tensor, not a vector, because for an alignment of the particle, head and tail do not matter. So you cannot describe it just by an angle or a vector. You need to go to higher moments. That's why you need a tensor here. So you have a derivative with respect to time and then also advection. On the right-hand side, you have the relaxation towards the minimum of the free energy. This is basically to giving the orientational elasticity of the particles. You penalize deformations in the form of bend and splay that are the dominant ones here. And importantly, we have the co-rotation term. This is because if you have elongated particle, not only this elongated particle moves with the flow, it has to also respond to gradients in the flow. So if there is vorticity, it has to rotate. If there is shear, it has to align to a shear. And that is characterized by this co-rotation term here. And then this equation for the orientation, this is coupled to the mass and momentum conservation. If I take this term out, these equations are exactly the same equations that people use for studying liquid crystals, nematic liquid crystals. You have in the stress, you have the viscous and elastic stresses. But now, because this system is active, we also need to take into account the active stresses that are being created by these particles. And the active stress basically takes this form of being proportional to the orientation direction. And this comes from the fact that we know that these particles individually, they create dipole-like flow fields to the leading order. And if you coarse grain that to the continuum level, you actually end up with a stress that is proportional to your nematic tensor. Physically, what this means is that since the divergence of the stress is going to create the flow here, any gradients in the orientation basically creates flow that moves these particles along. Now, the nice thing about the continuum theory now here is that we can take the configuration of the defect, impose it here, and then use the Green's function solution to find the corresponding velocity field. And what we find is essentially this analytical calculation that corresponds to both the discrete model and the experiments. And the important point is that this comet-shaped defect, because of its broken symmetry, basically has a self-propulsion property. It moves towards its head direction. And the more active the system is, it should move faster. This is a prediction. They go check it in the experiments. And indeed, if you compare the plus half defects in these comet-shaped defects in the fast and the slow ones, you can see in the fast-moving one from the MSD measurements, the plus half defect moves much faster in the fast-moving bacteria, while minus half defect, just to go back, it has this threefold symmetry which correspond to symmetric flow field, and it doesn't change its velocity when you make the particles more faster. Now, at this point, then we had the idea that what happens indeed is that in the fast-moving bacteria, the comet-shaped defects are faster. When they're faster, they can come closer to each other and effectively form this kind of Rosette structures that have very high energy. And from liquid crystal theory, we know that in a 2D layer, if you form this kind of a Rosette-like structure, you have the process of escape to the third dimension. So essentially what happens is that they stand up on their end. You can check this in our simple self-propelled rod simulations. We can shoot two defects to each other. And indeed, if we are doing this for fast-moving bacteria and look at the angle that they have with the horizontal plane, we can see once we shoot two defects to each other, they stand up, the rods stand up at their end, some kind of a reverse domino effect, and so they cannot expand further. What's more important is that we see the exact same thing in the experiments, that exactly when these two defects come together, the bacteria get out of focus. And indeed, in also the mixed culture, we can see that the fast moving ones are the ones standing on their end, and so they cannot expand further. So this basically tells that there is some kind of evolutionary speed limit on the motion of these bacterial colonies. If they're moving faster than a certain speed, they get trapped in the structures of their own creation. And we can actually understand quite a lot about this by using the theories of active matter, and in particular, active liquid crystals. This was the first example. And the second example, I'll talk about a completely different system, but maybe some kind of similar physics. And now what I'm going to do is that I'm going to look at a system of eukaryotic cells, in particular epithelial cells. And these are the cells that are lining up the surfaces and cavities in our body. And this is done, experiments are done by Suang Wensa and in the group of Benoit Ladoux. And what we find is that if we look at the elongation direction of these cells, we again find these comet-shaped and trefoil defects. And what happens is that the cells actually they die and get extruded at the position of these defects. When they check it for different experiments, for kidney cells, skin cells, aggressive, non-aggressive breast cancer, in all of them this happens. And under the microscope, these are essentially these white spots that you would see if you image a epithelial layer. And of course, again, this is an active process. These are continuously moving around, and the cells are being extruded out of a monolayer, which is an important process in order to keep the integrity of the tissues, as the cells also constantly go through divisions. And here you can see an example of cell being extruded, this white spot appearing at the head of these topological defects. So again, we wanted to understand what is so particular about this system that shows this extrusion and at the position of the head of the comet-shaped defects. Of course, again, we compare the flow fields, the shear rate, it matches between the continuum theory and the experiments. But more interestingly, in these experiments, they can measure the mechanical stress that cells exert on the substrate. So we can look at the isotropic stress, essentially pressure, and the blue one would show compression and red color would show tension. And here you can see clearly that at the head portion of a +half defect, both from experiments and from the continuum simulations, we have this high compression. And the theory was that this high compression is indeed responsible for pushing the cells out of the monolayer. Indeed, in the experiments, you can see that this negative pressure builds up before things being extruded. And basically what we did was to reformulate what happens in terms of cell death and extrusion. These monolayers, they behave like active liquid crystals. They continuously create these topological defects. If the cell happens to be at the head portion of defect, the +half defect, then the compression essentially kills the cell and almost simultaneously pushes it out of the monolayer. But if you think about it, this system is a little bit strange when you compare it, for example, to the bacterial system, in a sense that the bacteria has a given orientation, while for these cells, these are continuously changing shape. These do not have a constant rod-like shape. And indeed, we define the orientation in terms of the direction of the elongation. So the question is that how does this actual order and how do these defects appear in this kind of system? And to answer this, we again turn back to some kind of cell-based model based on the idea of treating these cells as deformable droplets, but active deformable droplets. What we do is to use a phase field formulation, essentially define a phase field that would take a value of, for example, 1 inside of cell and 0 outside. So there would be a diffuse interface in between. And this term on the right-hand side, this would basically be classic Can-Allen type equation that through the free energy, we can determine the interface properties like surface tension, we can have the cell repulsion and the elasticity of the cells. But in addition to this, we have a term proportional to the velocity for each cell that drives the system out of the equilibrium. This is the source of activity for these cells. And this velocity is determined from force balance for an overdamped system. Consider overdamped system. So the force also can be written as the divergence of the stress. So all we need to describe is this stress tensor. And the simplest way that we started with was to consider this stress to be proportional to the shape tensor for each of the cells. This shape tensor essentially would mean that the largest eigenvector, the eigenvector corresponding to the largest eigenvalue would give you the direction of the elongation. And physically, what it means is that if the cell gets a little bit elongated, then there will be some kind of a bootstrap effect that will elongate it further, and then this will elongate the neighboring cells and would drive the system out of equilibrium. This was the simplest possible way of doing it. We have recently done it. I mean, this is also this kind of dipole-like fields you can also measure in the experiments, and you can also put more complicated forms. I think we have figured out a more accurate way of doing it. I will not go through this one here, but let's just stick with this simple description of the stress proportional to the elongation, to the shape tensor of the cell. And with this, then you basically create again these collective patterns of motion. If you look here, you can see that the motion starts to appear as these cells push on each other and they move around. One can look at the corresponding velocity field that shows the emergence of the vortices and jets. But more importantly, we can measure again the pressure and the isotropic stress from the discrete model. We compare it with the continuum model, we compare it with the experiments, and they all match. Which is quite amazing, because I have now introduced to you two different systems, bacteria, epithelial cells. I have used the same continuum theory and two different individual based models, and all of them agree with each other in terms of describing at least the flow and the stress and the dynamics of topological defects. So for the rest of the talk, I'm going to then focus on this continuum model and see if we can do some sort of theoretical and numerical analysis on that to learn more about this system. And just to sum up this part, essentially what we found was that this high level of mechanical stress at plus half defects has a biological functionality in terms of killing the cells and extruding them out of a monolayer. So we can indeed use some of these physics ideas to learn about biology. And these studies on the defects, they have exploded since. We now know in human fibrosarcoma, for example, these defects, they pump the fluids when you put them in a confinement. In neural stem cells, these defects are the locations where the cells accumulate. And also in myoblasts, they have recently shown that they can form this very fascinating tornado-like structure. Just to say that these functions of these defects are continuously growing currently. So I hope by this point I have been able to convince you that these defects are important, and we can learn actually something about biological systems by using these kind of active fluid and active liquid crystal models. But there is one problem that with these defects in general. If you look at experimental systems, here for example, human bronchial cells or swimming bacteria, we see that these defects are actually do have actually kind of chaotic dynamics. They get continuously created, annihilated, and they move around in the system. And this is a phenomena basically known as active or mesoscale turbulence. That is a fascinating topic on its own. I have just listed some of the references here. I'm not going to go through active turbulence. What I'm going to do is to then ask a question that, okay, we know these defects are important. We know these systems show this sort of chaotic behavior. Can we do something to control these flows and to control the position of these defects? I will introduce 4 different strategies and walk you through those. So here is what happens if I simulate those continuum simulations that I showed you at the beginning. We get these chaotic flows appearing, we get these chaotic topological defects, qualitatively similar to experiments. One thing that you notice then is that these vortices that are appearing in the flow, they have a certain length scale. So what we asked was that, what if we introduce some kind of hydrodynamic screening? We do it simply by adding friction to the system. What friction does is to introduce a new length scale. As you increase the friction, the screening length scale goes down, and you would expect at some point it will become comparable to its intrinsic size of vortices. And indeed, if we are there, then the system self-organizes into a lattice of vortices. And more amazingly, then this lattice of vortices gets interleaved with an ordered array of topological defects. So basically, you get some kind of structural control over these patterns of motion. Now, based on this hydrodynamic screening, we can do another thing. I alluded to this a little bit in the introduction. So we can put this thing in a confinement. Here, then the confinement would introduce the screening length. And by varying the size of this channel, I can then change the amount of screening. So if the channel is small, or if the activity is small, you would get this unidirectional flow of the material inside the system. Essentially, the channel size is smaller than the intrinsic length scale of the vortices that the active matter wants to create. So it just gets streamlined across the channel. For larger channels or for larger activities, we get again these sort of chaotic flows. But again, we can go to a certain size where these vortices get arrested, and we get these beautiful dancing patterns of the vortices that are dynamically moving around. More interestingly, we see that the corresponding topological defects now show this sort of ordered state where, if you look closely, you can see 2 defects. They interact with one vortex and then move to another vortex and then go through the system.
Our next dance is one you'll definitely
So this reminded of this Cayley dance that you will see shortly here.
Cayley and Scottish country dancers. The first couple turn by the right hand at the top, usually a barrel at Cayley's. The first lady turns second man left hand.
So you get the idea. So what happens here is that now we have these topological defects interacting with the vortices. They change their partners, and by doing so, they basically span the entire system and show this ordered structure. This is very beautiful to observe numerically, but it was really amazing to see it also in the experiment recently from Francesca Gues's lab, by confining those microtubule motor protein mixtures and observing this sort of Cayley dancing dynamics of the topological defect. Now, in addition to being some kind of cute state, these vortex lattices, they seem to be quite important in terms of how these active systems, they transition into chaos. If we consider this vortex lattice and then the turbulence state, if I go somewhere in between these two, I can have a coexistence phase where I have ordered vortices and disordered patterns of vortices. I can quantify this by measuring entropy, average it over the height, and have a one-dimensional signal that would show the order and disorder. And so this is for one snapshot. That I can look at this over time, this one-dimensional signal over time in a so-called chemograph. Then you can see that these, the so-called puffs, they get created in the background of a vortex lattice as the systems become chaotic. And they can grow, they can branch out, and they can die. Now, the interesting thing is that at certain low activities or small channels, these puffs, they die out in the system. If the activity is too high, they would go through the entire system. But there is again a critical activity where this rate of branching and then rate of the disappearance, they balance each other. So you have, at any time, you have a certain fraction of your channel being occupied by these chaotic by these puffs that they characterize the disorder in the system. We can then measure the fraction that has become chaotic as a function of the activity, and we see that there is some kind of a power law growth. The important thing is that then the exponent is 0.276, which is exactly the exponent for the directed percolation universality class. And this is important because for the classic turbulence, when we have a transition from laminar to turbulent flow, this was shown in recent years that the transition to the classic turbulence actually belongs to the directed percolation universality class, both in 2D and 3D in independent studies. And what we are claiming here is that for these active turbulence, if you call it turbulence, these active chaotic flows, which are essentially happening at 0 Reynolds number, we also observe the directed percolation universality class, which sort of suggests that there's some kind of universality in terms of transition to this kind of chaotic flows, although the mechanisms and the mechanics is completely different. And we can now, everything I have told you what has been in 2D, we can just look at the 3D case, just consider 3D channel, now change the height of this channel. If the height is too small, we will have essentially quasi-2D system. The defects would be identical in the different planes, and the flows will be identical, and we will have these chaotic flows. But again, if we make the channel height larger, then the defects start buckling. So now these lines start buckling, and that corresponds to transitioning to three-dimensional chaotic flows that can be clearly seen by measuring, for example, the projected distance between the head and tail of these defect lines that would, and the curvature of the lines that would correspond to the emergence of the 3D flows. And indeed, what we find is that this happens because there's an inherent twist instability that twists the lines, and then you go from something that is completely 2D to something that is 3D. And this again has been now observed in the experimental systems in this paper in 2020 with the system of motor protein microtubules stabilized by FD viruses, that you observe again these three-dimensional loops and lines and the twist that I mentioned earlier. So, and the penultimate method I want to introduce in terms of stabilizing the system is through the pattern medium. This is collaboration with Oleg Lavrantovich and Igor Aronson, and brilliant work of Christian. And the brilliant idea of Igor and Oleg is that if you then put these bacteria inside a passive liquid crystal, by controlling the orientation of passive liquid crystal, you can control the orientation of bacteria. What we do in this study is then we design the patterns, this kind of a C-type patterns that you see here. So you have bend here, and you have some kind of a splay here. And when you put bacteria inside this kind of pattern in the experiments, you see that bacteria basically they concentrate in the regions of splay and get depleted from regions of bend. You can see this beautifully here, that when you put the bacteria, they form these very nice straight jets that over time, as the concentration of bacteria increases, they start developing these undulating instabilities, but the instabilities do not grow beyond the bend region. So it is undulating, it is moving, but it is not growing completely unstable. And so the advantage is that then if you have these kind of strong jets, then you can use it. For example, in these experiments, they use it for transporting cargo in real time, putting these microscopic colloids in the system that bacteria would move along, and they can also move it in these undulating patterns. So we came back to the original continuum model. We introduced, to explain this system, we introduced a binary order parameter to distinguish the regions of active and inactive. But the important point is that then in our system, we also introduce in the model a certain alignment to the background direction. So the direction not only it is described by this equation and the alignment of the particles, but also the particles have the tendency of aligning to the background pattern. if we do this, if we start, for example, with a slab of active system that is in yellow in a background that is shown by red, you can see that spontaneously this basically condensates into those display regions, similar to the experiments. You can also characterize the concentration, for example. And we can also see the same instability appearing and leading to these sort of undulating patterns that remain within the bounds of these bend regions. You can measure the amplitude, for example, and show that it plateaus. So again, using these pre-patterned surfaces appears to be a very, very strong, very powerful way of controlling patterns of this system. And the final method that I want to discuss is using topological constraints. And this is motivated by beautiful examples from Andreas Bausch's group. They take these microtubule motor protein mixtures and then make an emulsion of this into a droplet. Now what you have is you have a droplet. At the shell of this droplet, these active systems leave, and then this droplet basically behaves like a living system. It constantly moves around, it creates these protrusions. But if you want to really model this system, it's a nightmare because you have an active system, you have a dynamic morphology, and you have also hydrodynamic in the background. But I think we have recently found a way of doing this, essentially taking again the same continuum equations, but Luke's brilliant idea was to basically introduce the coupling to the order parameter such that the active system gets stabilized only at the shell and not inside one phase or another. And by doing so, then you get this kind of pattern from the simulations. Again, you can see this active material moving around. You can even notice those comet or trefoil defects being created and annihilated. The protrusions are appearing. And if you look more closely, you can see that protrusions appear actually at the location of the defects. And they leave behind these -half defects at the areas that you have the lower negative curvature. And of course, then with this, you can vary the activity of the system, how active these particles are, and get zoo of these different shapes that you can have on this sort of a bio-inspired material that I introduced. And at this point, I just want to mention a very beautiful experiment that has recently appeared and seems to also give further insights onto these dynamic morphologies and their applications in organ formation or morphogenesis. So what they do is that they look at the sea animal called hydra. These are essentially regenerating animal, because if you take any part, cut any part of this animal, it would then self-organize into hollow sphere. And if you wait another 2 days, it would go back to the original shape. You can see example here that this hollow sphere time here shows hours. And you can see after about 48 hours that initially hollow sphere basically goes back to the original shape of the hydra, which has a mouth here and the feet here, and then 2 tentacles that are appearing. So our idea was that these tentacles should appear at topological defects. And indeed, if they go in the experiments and they characterize the structure of the cytoskeleton, the actin structure on this animal in vivo, they found actually the mouth and the foot, they appear at the position of the defects, the full integer defects. The tentacles also appear at the defects. You even can show that you conserve the Gauss-Bonnet theorem, and you have these -half defects left behind as we expected from the earlier simulations that I showed you. And just before ending, this idea of using basically emulating nature to find certain type of fluids that they can self-deform or self-heal or self-organize seems actually not really new. People have thought about this maybe 30 years before us, as you see probably from this, you have seen probably from this Terminator movie. Again, we are not there yet, but you get the idea. With that, I just come to the end. And what I have done is essentially walk you through a couple of examples of how we can use these active matter theories to understand biological systems and introducing different ways of how to tame these kind of active materials. Thank you very much for your attention. I would be happy to take any questions.
I wondered if I could start the questions by asking you, what are the energetics of modeling bacteria? I mean, in terms of how the rate at which they put energy into the flow, is it modeled at a constant rate, or does it depend on how fast they're moving?
So what we do, at least in these very sort of simple models, is that this rate, we consider it as a constant.
Right.
And it comes essentially by considering this, if you think about the individual-based model, by essentially considering a force that is constantly acting along the head of the bacteria. Okay.
I see.
In a sense, then, you know, the system is always out of equilibrium. You are constantly putting entropy in the system. And the same thing happens with the continuum. The active stress that you have, that cannot be captured by any free energy formulation or so. It is a non-equilibrium term that we consider, at least in all these simulations, that food is always available to them. So there's constant source of energy. Of course, in real systems, when they run out of food, then they would stop.
Right. So it's a constant force model rather than a constant energy injection, because I guess it's a question of how fast the bacteria are moving, right?
Yes, I think at least in the context of the simple self-proper rods model.
Right. Just a follow-up question. So, you know, in non-active fluids, usually, you know, as a system becomes more complicated and it goes through bifurcations, there's usually an underlying trend that the energy dissipation in the flow is increasing, or perhaps decreasing, depending on how it's forced. When you look at bacterial turbulence, is there the same sort of ordering, given you're putting energy into?
Yeah. So this is the very good question, and this is indeed studied quite a lot by us and others in the context of this mesoscale turbulence, looking at, for example, power spectrum, as you do, for example, for the classic turbulence system. You don't have things like the Kolmogorov scaling or the cascades that you see, but you have some cascading effects in the sense that energy is being input at the level of individual particles, and it's getting dissipated at the level of these collective vortices that appear, but they have an intrinsic size. That is the main difference compared to the classic turbulent-like flow.
Right.
Again, I should really emphasize, I tried to emphasize it when I talked about directed percolation. These are completely different systems when you think about it. It's like zero Reynolds number, energy injection is local, and they are different in many senses. I'm not sure if calling them turbulence is correct, but they show some similarities with the, at least in terms of transition to chaos.
Great talk. I mean, I really enjoyed it. I have two questions. They're related. The first is, what are the conditions for creating such topological defects in terms of individual swimmers? Let's say, is the anisotropicity of the rods an important factor or not? And the second question is that, would your model be able to predict some defects in synthetic active matters, such as the Quincke rollers of Bartolo group? I'm very excited to know whether this is possible or not.
Yeah. So for the first question, the conditions for the defect nucleation. This essentially goes back to seminal work of Simha and Ramaswamy, where they looked at these hydrodynamic instability that would happen in these active systems. But in order to have the topological defects, we need to have some kind of, at least this type of topological defects that I'm talking about, you need to have the orientational order, right? So you need to have some orientational order that can allow singularities. Once you have the orientational order, if you start from completely aligned states, it would become unstable. Let's say first to this, you can even see it in this video here, you can see this bending pattern. Would get unstable to bend the structure. Then these bends, they have high elastic energy. So the system gets rid of them by nucleating two defects that unzip this bend region. Okay. So in the context of these sort of nematic topological defects, you need the orientational order. And I gave also the example of the epithelial cells, which they look circular, but they are able to change shape. And if you look at now the anisotropy in their shape, then you also find topological defects. With the Quincke rollers, I'm not sure you will see this kind of topological defects because those are really circular in shape and they are rigid in terms of shape. There's no shape changes, but you certainly can observe, for example, dislocations in those systems. The other possibility is that if you look at the velocity field of those systems, because these Quincke rollers, essentially they have a polarity direction. Polarity is a vector field. If you look at that vector field, you are going to have again singularities in terms of the full integer topological defects. You are going to have essentially things like asters or vortices that I showed earlier.
Yes.
Yeah, my question is related to the first and the final slide in the presentation. So is it interesting to study the vorticity distribution regarding the transition to active turbulence and for studying the matter? And could it contribute towards the redevelopment you showed in the final slides?
So I didn't understand the second part, but let me address the first part, then maybe I ask you to repeat the second part. So in terms of the defect distribution, yes. So we and others have looked at the defect distribution. Indeed, there's a very nice work from Giomi in PRX 2015 that he analytically also looks at the vorticity distribution in these sort of active systems, and he predicts some kind of exponential decay and a certain size for the vortices that have also been in 2019 also probed in the experiments on microtubules by Francesc Sagues's group in Barcelona, and they show that you actually observe that kind of distribution. So that's the first part. So yes, indeed, you can look at the vorticity distribution. I don't know if you would learn things about the transition that would be interesting to think about. And the second part, if you can see it again.
Yeah, sure. The second part is, as you explained in the final thing, that the future scope is we will try to redevelop things we will try to redevelop the active matter as from the illustration in the movie. So can the vorticity distribution, the rotation of the vortices, could that help in that redevelopment, if I may say it?
Yes, sure. The idea is basically, I think also most of the things that I discussed, they were based on the fact of somehow arresting that vorticity length scale, which we know, at least for these active systems, it is also tightly connected to the topological defects. So for sure, if you can control the vortices, you are going to also control the defects. So one of the things, for example, we have done, I didn't talk about it here, is putting disks inside these active systems. These disks are pinned in place, but they can rotate. So essentially, by doing this, you will have vortices moving around the disk, and you would get something like anti-ferromagnetic order. And similar studies have been done by considering, for example, different cavities and putting these cavities next to each other in different configurations from Ray Goldstein's group. And then you can see that the bacteria self-organize again to the anti-ferromagnetic or ferromagnetic order based on the structure of the lattice that you are imposing. So for sure, yeah, if you can control vorticities, you're going to have also some control over the material.
Is there a clear separation between chaos and turbulence in these systems?
No, again, I would not call these systems turbulence. and they are chaos in a technical term, that they really depend on the initial conditions and so on.
Right.
But I actually don't know how you would distinguish chaos from turbulence in these systems.
I guess, I mean, how, you know, what's the separation between the largest length scale of your system and the actual bacterial size?
So the length scale of the collective flows that you observe can be order of 100 times larger than the scale of individual bacteria.
I see. Okay, fine. Right. Let's see. So Ayush asks, how do you perform the simulations?
Okay, so for doing these simulations, we use lattice-Boltzmann method to model the fluid. So we have this kind of hybrid lattice-Boltzmann. So lattice-Boltzmann models the fluid, and the Q tensor, we use a simple finite difference. And in the case that we have phase field, we also use simple finite difference for that.
Right. Okay, next question. There is some kind of analogy with the pair creation and annihilation of particles, antiparticles on one hand, and the pair of comets, trifoils on the other. Can we test some QFT predictions with active matter?
That's a very tempting question. I can tell you that there are some talks also, even, you know, using these ideas of active matter to look at sort of cosmological problems, because again, at the early universe, there are some theories that what happened was that you essentially nucleated the defects, and of course that was a really, really active system. I don't know if you have ideas, you might give it a try. Of course, the nature of the thing is very different when you are thinking about the quantum systems, because over there you are describing the system with a Hamiltonian. Here, Hamiltonian really doesn't work because the system is really out of equilibrium. But yeah, well, one never knows, I think.
Okay. The next one is from Tulio. I have a question about the formation of the defects. I wonder if you could unmute yourself.
Yes.
Tulio.
Can you hear me?
Yeah. Yes. Go ahead.
Thank you for a great talk. It's a question about the formation of the defects. I was wondering, basically, so as far as I understand, you can simulate these models with continuous equations. And for example, you can have the instability of the aligned state, which leads to a state where there are defects. And I was wondering, is the steric interaction, in the sense of the exclusion of volumes, coupled with the fact that these are rods, necessary to have these kind of defects, or sort of can one imagine the formation of these defects from interactions that are more far field, so not steric?
That's a great question. I think you can have the defects in both scenarios. And when you think about, for example, the cellular system, they have a very different type of interaction compared to the bacteria system that at least we thought it was some kind of repulsive interaction. In cells, they actually form very strong cell-cell junctions. There are certain families of proteins that essentially they put the walls of two cells, they knit walls of two cells together. So they stick together. So there is some kind of attraction, adhesion, and also deformation of the interaction. And still in those systems, you observe the topological defects. And as I showed, when you use the continuum theory, that is basically just in terms of putting the source of energy and considering the penalty for any orientational deformation, whether the source is because of the repulsion or attraction or thing doesn't matter, you get the topological defect. So in that sense, it is, I think it's quite generic. So even with the far field interaction, you would also observe the topological defects. And again, and one other point I should mention, so these are, the reason they are called topological is that you cannot really remove them by any smooth transitions or anything. They are just imposed by the topology of the problem. So I think the conclusion will be generic also for the case of having different types of interaction.
Thank you very much.
How do you actually choose the simulation parameter, especially when you want to directly compare your simulation research with the biological situation? I guess I wonder how you extract the parameters from the observation, or I don't know if there is any way to do it.
That's again a very good question. I think when we start, that is still one of the challenges. I think, to me, one of the future challenges of active matter is to be able to do more quantitative analysis and comparing with experiments. When we started, we would just consider, for example, generic values and then look at the patterns. We have done more quantification, I would say, recently in terms of, for example, trying to match the emerging length scales and time scales that appear in the system. But again, this really comes down to the question that you are asking. For example, if I'm asking about a epithelial cell, right? I know inside the epithelial cell, there are millions of genetic pathways going about that. Of course, there's no hope of capturing those in a model, but I know those millions, they give rise to a handful of mechanical properties. So I can start looking at those mechanical properties like the speed or the elasticity of the cell, and then compare those and build essentially dimensionless groups and compare those with what you get from the theory to get some mapping between the parameters. But overall, this is an active field of research still.
I see. So basically, okay. Yeah, thank you very much. Anyway, yeah.